Okay, before we begin, are there any questions? Actually, some people are still logging in. Not sure what that's about. All right, I'm not hearing any questions, so can somebody confirm that you can hear me? Please?
Yes,
Yeah.
we can hear you.
Thank you. All right, so today is September 28th, and we are going to continue Chapter 1 and Chapter 10. There is a lab today. Let me remind you that lab 00 must be turned in by 8 a.m. this Friday. Normally the lab worksheets and the lecture worksheets are due at 11:59 p.m. on Saturday, but lab 00 I need to get in a little earlier so that I can determine who is not attending the class and then drop them. And I think I have to do that sometime on Friday. It used to be 5 p.m., but I think they moved it up. All right, any questions about what we're doing? All right, I had marked that this is where we ended. We were talking about what a cell is, what's in a cell. So here in these pictures, where would we not find a microorganism? Would we find it on the plants and in the water and maybe the soil there or the rock? Yes or no?
Yes.
Okay, we would find it in the ocean water, and we're talking about 1 cubic centimeter.
Yes.
Yes. How about in the air above the ocean? Would we find microbes?
Yes.
Yes. Actually, we find microbes up about a mile into the atmosphere, and that's because the wind can pick up spores and small microbes and then carry them. The same within the soil. We can find microbes in the earth down about a mile. And that's true whether we're talking about the soil or we're talking about underneath the oil, underneath the ocean, meaning underneath the, what do they call that? The bottom of the ocean. As long as it's not rock, we can find microbes in it. And even in some rock, we can find microbes, but obviously if this was solid rock here, the microbes couldn't get through it. And then we find microbes on the person and on the cat. We find microbes in the desert and in the desert air. We find microbes everywhere except for one place in this picture. This is a magma vent or a lava vent, and you can see the magma right there. And that's the only place we would not find microbes. We would find microbes on the rock here and even here, but not where the lava is. It's too hot, and that would destroy any microbe that fell in there. So all the places you'd find microbes, where you would not. So microbes are found almost everywhere. And by everywhere, we mean any environment where the land and the water, atmosphere, subsurface on the land and the ocean, and on plants and animals, essentially anywhere where water is present. So we can find microbes growing and living between temperatures of a minus 20 degrees Celsius and about 120 degrees Celsius. below a -20, you might find frozen microbes, but they would not be growing because they would be frozen. And some microbes can survive freezing temperatures and would thaw out fine. It is true that if you freeze bacteria about and then thaw them, about half of them will die, but half of them will remain alive. That's not the case with the eukaryotic cell. If you freeze a eukaryotic cell, the water crystals will kill the cell. But with bacteria, about 50% of them could survive freezing and thawing. It does depend a little bit on the species. Endospores, which we'll talk about more later in the class, would survive freezing. They'd survive freezing quite fine. And then, like I said, microorganisms can go up to 120 degrees. The record, which may not be the record anymore, because I read this, boy, it might have been 20 years ago. It might have been 15 years ago. It was a long time ago. They found a microorganism that could survive at 126 degrees Celsius. And it turns out that under the Marianas Trench in the Pacific Ocean, the deepest part on the planet, they found a microbe in a volcanic vent in the Marianas Trench, and it was living or surviving at 126 degrees. Above 126 degrees, you would have water boiling. And I suppose a microorganism cannot survive when the water within the microbe boils. Any question about any of that? All right, let me warn you, somebody's got a microphone that's on. If you could mute that, that would be good. Microbes are numerically the most abundant life form on the planet Earth. How many microorganisms are there? We're talking about prokaryotic cells. In 1 gram of soil, we have about 1 × 10⁸ bacteria and archaea. And I suppose that might be yeast as well. But the point is cellular microbes. If we were to talk about viruses, it would be about 10 times that. And we're just not going to talk about microbes now. We're only going to talk about prokaryotic cells. It does depend on the moisture in the soil and the amount of organic matter in the soil. Generally, the more moist the soil is, the more organic matter it is, the more microbes you'll find. And then the drier the microbes and the less organic matter you'll find, the less frequent the microbes would be. But even in the desert, which the soil is obviously very dry, and we usually call desert sand. We don't call it soil, but it's the same essential thing. And we do find lots of microbes there. And there's very little organic matter there in the desert, sand or soil either. And so we find at least 1 × 10⁶ microbes, but generally in most soil, we have around 1 × 10⁸. In a milliliter of ocean water, which would be 1 gram of ocean water, do we have more or less microbes than 1 × 10⁸? I'll warn you, this one is probably the hardest of all of them. Anyone have a guess, more or less? You got a 50-50 shot here.
Less.
You're right. Ocean water is considered one of the deserts on the planet, and you have less microbes in ocean water than you do in normal soil. In a gram of human feces, do you have more or less than a gram of soil? This one isn't difficult. I would think most people know that. Yeah, you have more.
More.
At least in most human feces. It does depend, obviously, how dry the feces are, but a fresh feces, which has lots of moisture in it, actually over 50% of the weight is from microbes. On a human being, of course, you have more. And of course, on the entire earth, you have a lot more. Let's take a look at some of the numbers. In ocean water, you only have about 1 times 10 to the 6 microbes per milliliter or 1 gram of ocean water. And I suppose 1 milliliter of ocean water is a little bit more than 1 gram because ocean water has salt in it, and that would add to the weight. So I was thinking it's about the same, but that's actually true only of fresh water. 1 gram of soil, about 1/17 to the eighth. We already said that. And feces, if it's very dry, like somebody poops in the desert and then it dries out, you'd have about 1 times 10 to the 6. But that's very, very dry, human feces. Most human feces have about 1 times 10 to the 11th cell in 1 gram. And as I stated, most of the weight of a feces, 50% of the wet weight, is from microorganisms. So an astounding number. On a human, we have 3.8 times 10 to the 13th. If I can read that right, let me blow this up so I can have an easier time. Yeah, well, 3.8 times 10 to the 13th. On the planet Earth, we have 1 nonillion. Those of you who have never heard of a nonillion, it's 1 followed by 30 zeros, or 1 times 10 to the 30th. An astounding number. For the human population, we're actually very close to 8 billion. We're just a little shy of 8 billion. So that's a comparison. A lot more microbes on the planet than people. When we're talking about microorganisms, you should realize that some of them do cause disease. Any microbe that can cause disease, we call it a pathogenic microbe. So a pathogen is something that can cause disease. And usually we're talking about a human disease. If it's ever a microbe causing another animal disease, we'd say something like a bird pathogen or something like that. But when we say pathogen, we usually mean human pathogen. Most microorganisms do not cause disease. Only a small percentage of all microbes are pathogenic. Only a small percentage cause disease. Any question about any of that? When we're talking about disease, it's important to distinguish 2 types of diseases. There are infectious diseases, and those are diseases that a person can catch, and inherited diseases. These are diseases that a person is born with. Infectious diseases are caused by microorganisms or a worm, which we'll talk about later. We do group them as a microbe, even if it's the adult can reach, what is it, 30 meters? The adult human tapeworm, for example, can be 30 meters long. An inherited disease is one that's caused by a person's defective genes. What's an example of an infectious disease? Anyone? You can be thinking, I'll ask in a bit about what's an example of an inherited disease. You guys don't.
Tuberculosis infectious?
Tuberculosis is an infectious disease, yes, caused by Microbacterium tuberculi, something like that. What's an example of an inherited disease? What's a disease that people are born with?
Huntington's?
Yeah, Huntington's disease, sickle cell anemia. All of these are inherited diseases. An infectious disease is one that's caused by a microbe. We have different categories of infectious disease. If it can spread from person to person, it's an infectious disease that we call communicable. If it's an infectious disease which is generally not communicable, meaning it will not spread, then we just call it a non-communicable disease. An example of an infectious disease that's non-communicable is botulism. If you eat canned food that has Clostridium botulinum in it, it can give you botulism, and generally that will not spread to another person. In fact, the only way I can even think of that spreading is if someone were to eat that person and undercook them, because cooking would even kill the botulism. But that would be a really rare case of botulism spreading. So generally, non-communicable diseases do not spread. Microorganisms, how small are they? Well, we got some pictures here. Let me blow this up. Most bacteria are on the size of about 1 millimeter to 2 millimeters. So that would be about the size of most bacteria. That's a little bit longer than most bacteria. This is showing you some fungi, and this is quite a bit bigger. Let me see if I can blow that up. So that's 50 millimeters, micrometers, and that would be one cell here, and that would be one cell there, and that would be one cell there. Here we have what looks to me like a white blood cell, and this is 10 micrometers here, so this would be about 50 micrometers, maybe 60 micrometers long. Here we're seeing a microbe, and this is unusual because this microbe is colonial. Actually, each of these dots is a cell, and this is the parent colony, and this is Volvox, and what it is it's made some baby colonies, 6 of them, 1, 2, 3, 4, 5, 6, and this will open up, and then the babies will go out and colonize other areas. Many microbes are singular cell, but there are some which are colonial or multi-celled, and Volvox is one of the few which is multi-celled. And I think that's a T lymphocyte, and then it's showing you the HIV virus right there, which is a microbe, but a virus is not a cellular organism. It's not a prokaryotic cell. Any questions about any of that? How small microbes are? They come in various shapes and sizes. What?
Can you repeat what you said about after you said that some are single cell? What did you reference the ones that are, did you say that they're colonial? Is that what you said? Colonial?
Yeah, most microbes that are not single celled are colonial. Like this one here is colonial. They're not highly organized into tissues and organs like the way most eukaryotes are, like all animals are. For example, you could, well, I don't know about Volvox, I just don't know, but you might be able to take off one cell here. And then if you give it in the correct environment, it can reproduce and then reestablish the colony. And that's what we mean by it's colonial. So I don't really want to get into that, but all bacteria and archaea, we say, are single-celled because even if they grow in a colony, if you break off one cell and then it were to grow, it'll grow the colony back. So we actually say bacteria and archaea, all of them are single celled. And it is true that some of them do grow in colonies, but one single cell can form the colony. With Volvox, I'm not sure. It's colonial, that's for sure. But I don't know if one single cell can grow back the colony. I just don't know. All right. We did mention last time that microorganisms play some beneficial roles on the planet and for people. They are important for decomposing organic waste. When something dies, it's mostly microorganisms that decompose it and then make the nutrients and elements available for other organisms, meaning they're the ones that are responsible for recycling the nutrients and the elements. It is true that there are other organisms, mainly fungi like mushrooms, which can decompose organic waste, but the majority of it is done by a microbe. Microbes are also producers in the ecosystem. They can engage in both photosynthesis and chemosynthesis. And as I mentioned, microbes actually make about 50% of the oxygen we have currently in the Earth's atmosphere. Green plants only make about 50% of the oxygen. So microbes are very important producers for the planet. We'll talk a little bit more about chemosynthesis later, but this is another way that a cell can fix carbon and make glucose. They can do that with photosynthesis or with chemosynthesis. Microbes are involved in the nitrogen cycle. We would not have nitrogen recycling on the planet if there were not microbes. Microbes also produce a large number of things for us, such as industrial chemicals. Ethanol and acetone are two chemicals that are typically made by microorganisms, and then the company that's making them will purify it and then sell the product. Like ethanol, they usually have yeast making it, and then they purify it and send it and add it to your gasoline. To my knowledge, they don't have microbes. Well, actually, yeah, the microbes do make beer and wine. Never mind, I won't mention that. I was thinking we don't have microbes turning corn into something you drink, but we do use microbes to make beer and wine, so you do drink it. We use microbes to make insecticides. These are naturally produced insecticides, which are an alternative to man-made insecticides. We use microbes to make fermented foods. All fermented foods use microbes to make them. And some examples of fermented foods are vinegar, cheese, bread, at least leavened bread, beer and wine. Even chocolate has to be fermented. We use microbes to make products used in manufacturing, for example, cellulase. We use microbes to make antibiotics, like penicillin G is made by a microbe. And then we use microbes to produce other products used in medicine. For example, human insulin is now made in microbes. You guys probably don't know this because this is a long time ago. When my father first started taking insulin because he was a diabetic, they first had to purify it from another human. So they were actually, don't ask me how they got this, but they were getting insulin from another human. And that just wasn't enough insulin for all the diabetics. So they then started purifying it from sheep. And my father switched to using human insulin to lamb insulin. And then he switched again to insulin that was made by bacteria. And what they did was they took the human insulin gene out of a human and then put it into bacteria, had the bacteria make the insulin, and then isolated it, and then gave it to the diabetics. Any question about any of that? All right. Microbes are also important in biotechnology and in genetic engineering. We'll talk about both of these. Biotechnology is the use of microbes and organisms to produce foods and products for human use. Biotechnology is not something new. It is centuries old. For example, when people make sauerkraut, they're using microbes to make the sauerkraut, and that's centuries old. That's at least as old as the Middle Ages. It may even go back much further than that. And that's biotechnology. We've also bred corn to highly produce corn oil. And the last I heard, we had corn that was 80% the weight was corn oil. They may have increased it, but I'm not sure. I guess the data I'm referring, that data is probably over 20 years old. It may even be over 30 years old. Anyways, that's biotechnology. Genetic engineering is a new technique of biotechnology. So when we're talking about biotechnology that's new, we usually mean genetic engineering. And that is usually taking out the genes from something, like a person, and then putting it into another organism, usually a microbe. Some applications of genetic engineering allow us to produce a variety of proteins from bacteria and fungi. Oops. Oh, that's right. Such as producing vaccines and enzymes. Another technology of genetic engineering is gene therapy, where someone is born with defective genes. You can take out, if it's a immune defect, you can take out their immune cells and then put in the correct gene in their immune cells, grow them up in tissue culture, and then put them back into the patient. That is gene therapy, where we can correct the person's genetic defects by adding genes to their cells. And to my knowledge, we only have this working in people who are born with a defective immune system. We can take out the immune system cells, meaning the B and the T cells, and correct them and put them back into the patient. Any question about any of that? All right, another genetic engineering is when we genetically modify bacteria, such as change the bacteria to help protect the crops from insects or from helping protect the crop from freezing. I'm sure you're familiar with the tomato saver tomato. How did you say that? Tomato saver tomato, which was a genetically modified tomato, which did not spoil as rapidly as tomatoes normally do. And the stores really like them because they could hold them on their shelves and the tomato would keep better. But apparently not very many people bought them because I haven't seen the tomato saver tomato or whatever it was called. I haven't seen that for many, many years. It used to be in every supermarket. Any questions about biotechnology and genetic engineering? All right. Microbes are important in human welfare. They're also important in the microbial ecology. Microbial ecology is microbes living together, creating an ecosystem. Microbes are important in recycling carbon, nitrogen, nutrients, sulfur, phosphorus. And because they do this, all of these elements and nutrients can be reused by plants and animals. We mentioned that microbes recycle nitrogen on the Earth's ecosystem. Well, they also help recycle carbon, sulfur, and phosphorus. It is true that microbes are not as important in the recycling of carbon, sulfur, and phosphorus as they are in nitrogen, where almost every step in the nitrogen cycle happens because of microbes. For the others, carbon, sulfur, and phosphorus, microbes are only acting at some steps, like phosphorus and sulfur can come out of rocks by erosion. The microbes have very little to do with that. And the same thing that the soil and whatnot can compact together and reform rock, meaning the sulfur and the phosphorus can rejoin rock. The microbes don't play any role in that part. So there are some steps in these cycles that microbes do not accomplish, but they are important in the cycling of those nutrients. And then we mentioned that when anything dies, it's mainly microbes that recycle the nutrients. The only exception is that some fungi, especially mushrooms, can also recycle the nutrients. And these that we've mentioned here are actually examples of microbial mediated biochemical cycles that support life on the planet Earth. These microbial mediated biochemical cycles were actually discovered by Winogradsky and Bajorink in the 1880s to 1892. They were the first to show that microbes were important in the nitrogen cycle, and I think it was the sulfur cycle. I think those were the first two that Winogradsky and Bajorink studied. And why they were able to find this is that Winogradsky and Bajorink, who were working together, by the way, did not study pure cultures. Normally in microbiology, we will isolate a species and then work with it in a pure culture, so that if the tube turns yellow, we know that it was that one species of pure culture that did it. Because if you have multiple species in the tube and then the tube turns yellow, you don't know which of those species or all of those species was the one responsible for turning the tube yellow. So in microbiology, we normally use pure cultures and only study pure cultures. But Winogradsky didn't do that. And he studied mixed cultures of microorganisms. And that is why he was able to see the nitrogen cycle, because he had lots of organisms in there, and different organisms were recycling a different nitrogen component to a different component. Any question about any of that? All right. Microbes are important for human welfare, such as remedying environmental problems. Bioremediation means literally life, bio, and then to remedy. The use of living organisms to deal with an environmental problem. Like these men here are spraying this oil slick with microbes so that the microbes can degrade the oil and then recycle it and change the oil to carbon dioxide and water. We can use microbes to take care of sewage. We've already talked about that. Why sewage is degraded is by microbes. Various pollutants like this oil spill can be degraded by microbes. And then even some things like mercury, we can use bacteria to treat the mercury and then make the mercury a less toxic compound to humans. So it still will be toxic because it is mercury, but you change it from like metallic mercury, which is very hazardous to humans, to an organic mercury, which is less hazardous, although it is still toxic to humans, but not as toxic as metallic mercury. Any question about any of that? Microbes are important in human welfare for other reasons, like microbes can make biological insecticides. These microbes are pathogenic to insects or the compound they make, and there are alternatives to a chemical pesticide. We can use these biological insecticides to prevent insect damage to crops and prevent disease transmission, mainly disease of crops. An example is Bacillus thuringiensis in this bag here, which is fatal to many insects. And yet Bacillus thuringiensis is harmless to humans and other animals and plants. My father, way back in the 1960s, treated, I guess that tells you how old I am, but I was just a young boy at the time. He actually treated his apples with Bacillus thuringiensis. And the apples had less worms than if he had not treated it. The trouble with the Bacillus thuringiensis is you have to apply it every 2 weeks. And that's actually the same as the chemical insecticides. You have to apply it every 2 weeks to the apples, or else the apples will get worms. And my father was way too lazy. He only did it one time. And then to make matters worse, he lived in Western Oregon, and Western Oregon and Western Washington have a lot of rain, and the rain will wash the Bacillus thuringiensis off the apples. So you must treat with Bacillus thuringiensis every 2 weeks, as well as after every rainfall. Any question about any of that? But it did work in the sense that the apples were less wormy than if he never had treated with Bacillus thuringiensis. And to get it worm-free, he just needed to do it every 2 weeks or after every rainfall. All right, any questions about microbes and their importance to humans or to the planet? If not, let's move on to the naming and classifying of microorganisms. Both the naming and the classifying of organisms started by this man here. Does anybody know his name? Who started the scientific names and the scientific classification? Nobody knows. It's Carolus Linnaeus, and your textbook calls him Carl Linnaeus. I think his Scandinavian, I think he was Swedish. His Swedish name was Carl Linné, but he loved Latin so much that he Latinized his name and changed it to Carolus Linnaeus. He established the scientific nomenclature where each organism has 2 names. He names each species with its genus name and its specific epithet name. For example, Homo sapiens is the species of humans, and Homus is the genus, and the specific epithet is sapien. You cannot use sapiens to name humans because that's the specific epithet name, not the species. The species is Homo sapiens. Any questions about any of that? All right. His nomenclature is so useful that other people started using it, and it is still the nomenclature that we use today. It is called the scientific nomenclature or the binomial nomenclature because all species have two names. Binomial means two names. His system of classification we no longer used. He classified all living things into plants and animals, and we don't use that classification system anymore. But he's the first one to start classifying organisms as well as naming them. Any question about any of that? All right, let's talk a little bit more about the binomial nomenclature system. The names are italicized or underlined. So whenever you type it, you should italicize the scientific name. If ever you're writing it like on a blackboard and you can't write italicized, you then write it out and then underline it. The genus name is always capitalized and the specific epithet name is always lowercase. The names are Latinized and they're used worldwide. So wherever you are in the world, whatever language you're speaking, you would always use the name Homo sapien to discuss a human, meaning that's the scientific name everywhere in the world. Any question about any of that? The name may be descriptive or it may honor a scientist. Some examples, we all already mentioned Homo sapiens in italics or Homo sapiens underline. Literally, this means ape or human that walks on two legs, Homo, and then sapiens, or is that wise man? Sapien is wise. Oh, Homo is man, sorry. So Homo sapiens, wise man. And something in there I think does mean walks on two legs. Escherichia coli is descriptive in coli because it grows in our colon. And then it's named after Dr. Astor, a early microbiologist. So this is honoring someone. Staphylococcus aureus is descriptive. Coccus means it's a round cell, like a ball. Staphylo meaning it grows like a cluster of grapes. And aureus means golden. So it is golden round cells that grow in a cluster of grapes. Any question about any of that? All right. After first use, the scientific names may be abbreviated with the first letter of the genus and then a period, and then the specific epithet spelled out. For example, Staphylococcus aureus can be used the first time as Staphylococcus aureus, and then all other times in your paper, you can abbreviate it as S. aureus. Both Staphylococcus aureus and E. coli are found in the human body. E. coli should be written as Escherichia coli the first time, and then all other times as E. coli. However, E. coli is so well known and used so much that oftentimes, even among scientists, they will write E. coli the first time. And that's just because it's sort of an exception. It should be this way, but the exception is because it's so well known, people just call it E. coli. All right, any question about the scientific nomenclature? If not, let's move on to the classifying of microorganisms. All organisms can be grouped into a larger group, and then organisms within that larger group grouped into smaller groups. Before we begin the topic of classifying, we need to look at the classification of all living organisms on the planet Earth. When we talk about all living things, we first classify them into the 3-domain system. The domain bacteria, the domain archaea, the domain eukarya. This is also sometimes called the tree of life because this looks like a 3-trunk tree. And then the trunk has branches where this organisms branch off of it. The domain is classified based upon genetic data, meaning we have a domain defined. And how you know which domain you are in is you simply sequence the DNA of the species, and all of the species within one domain will be more similar to each other in the sequence of their DNA than any other organism in another domain. So if we were ever to find a new species and somebody wanted to determine which domain does it go in, all you need to do is sequence the DNA, and then that will tell you which domain it belongs to, because its sequence of DNA will be more similar to the bacteria or the archaea or the eukarya than any other organism in another domain. Any question about any of that? So the domain is defined. We know exactly what each domain is. You just sequence the organism and then can determine which domain it belongs to. There is no argument about which domain an organism belongs to or how many domains there are. Any question about any of that? We can then group organisms in smaller groups underneath the domain. And the first grouping we're going to talk about is the kingdom. The kingdom is not defined. There are arguments as to what a kingdom is, how many kingdoms there are, and which organism belongs to which kingdom. We're going to use the 6-kingdom approach, but you should realize that some people use a 3-kingdom approach, some people use a 5-kingdom approach, probably the most use a 6-kingdom approach. Some people use a 9-kingdom approach, others a 15-kingdom approach, others a 20-kingdom approach, and others some number above 20. Okay, like I said, the number of kingdoms there are and what a kingdom is open for debate. And scientists have been arguing about how many kingdoms there are for over 20 years now. So they're not going to decide this anytime soon. We will use the 6-kingdom approach, where the domain bacteria is also the kingdom bacteria. So the kingdom and the domain are the same. And the domain archaea is also the kingdom archaea. So the domain and the kingdom are the same. The domain eukarya has 4 kingdoms in it, meaning this will be the 6-kingdom approach we're going to use. Kingdom bacteria, kingdom archaea, and then kingdom plantae, the plants, kingdom animalia, the animals, kingdom fungi for the fungus, and then kingdom protista for the protists. Any question about any of that? All right, remember the number of kingdoms is open to debate. We're just using the 6-kingdom approach because it works well with the domains. And if you have more than 6 kingdoms, what you do is you just divide the kingdom Protista up until at least about the 20 stage. After the 20 stage, you may divide other kingdoms up, and you also divide the bacteria and the archaea up too. The kingdom Protista is not a monophyletic group. The kingdom Protista is a eukaryote, meaning a eukarya, that is not a plant, animal, or fungi. So the kingdom Protista, we always knew, was not monophyletic, meaning it did not have one common ancestor. It was not one, what's the word I'm trying to say, one grouping. We always knew it was multiple groups together by its very definition. A eukaryote that's not a plant, animal, or fungi. Any question about any of them? All right. When we're talking about the 3-domain system, your textbook says that they can have one of 2 cell types. And they call the cell type a prokaryote or a eukaryote. I should warn you that different authors don't call it a cell type, they call it a super kingdom, meaning you can divide all living things into a super kingdom, and then you can divide the super kingdom prokaryote into the bacteria and the archaea. Obviously, the super kingdom eukaryotes is the same as the domain eukarya. And we're not going to talk about the super kingdom. I'm just warning you that the cell type is what other authors call it. They don't call it a cell type. They'll call it a super kingdom. But we will go with the cell type because that's the way our textbook author uses it. The cell type for bacteria is prokaryotes because they have prokaryotic cells. The cell type for archaea are prokaryotes because they have prokaryotic cells. And then eukarya, of course, has eukaryotic cells. All right, any question about any of that? No, I can't go back. There we go. So let's talk a little bit more about the domain bacteria. Their cell type is prokaryotes. You can find peptidoglycan in the cell wall of all bacteria. When bacteria divide, they mostly divide by binary fission. For their getting energy, bacteria can get their energy from all possible sources. They can get their energy from an organic chemical such as glucose, and that's how we get our organic, how we get our energy from an organic chemical. They also can get their energy from an inorganic chemical, something like iron, a sulfur compound, or a nitrogen compound. These would be the chemosynthetic bacteria, and we'll talk a little bit more about them later. And they can also get their energy from photosynthesis. These would be the green or the photosynthetic bacteria. Most of the bacteria are either free-living decomposers or primary producers. But some of the bacteria are parasitic, and some of them can cause disease, so we say they are pathogenic. Any question about the bacteria? The domain archaea, they do have prokaryotic cells. They do not have peptidoglycan in their cell walls. Some of the archaea are found in extreme environments. And so if you have an older version of the textbook, you'll find that the author tells you that archaea are found in extreme environments, and period. They won't discuss further. In reality, only some of them are found in extreme environments. in the last 15 years, we've looked at other environments and we found that you can find them in all of the biomes on the planet Earth, meaning that they are everywhere. They can be found in methane environments, the methanogens. They can be found in extreme salt concentrations like the extreme halophiles. They can be found in extreme temperatures, the extreme thermophiles. But wherever we look on the planet Earth, we'll find the archaea. They actually are a significant portion of the life in the ocean, part of the plankton, and they are also a significant portion for the microbes in the soil. Any question about them? All right. For the archaea, none of them are human pathogens. None of the microbes are pathogenic. So after this lesson, we're not going to be talking much, if at all, about the archaea. And the main reason is not because they're not important, not because they're not interesting, but simply because the focus of this class is going to be on discussing human pathogens, and none of the archaea are human pathogens. So we're just not going to talk about them much after this lesson. Any question about the archaea? All right. For the eukarya, remember they have 4 different kingdoms: kingdom Plantae, kingdom Animalia, kingdom Fungi, kingdom Protista. All of these cells in the eukarya lack peptidoglycan. So peptidoglycan is only found in the cell walls of bacteria. And we'll talk more about peptidoglycan later, not today. All right, let's talk about the kingdom fungi. Fungi is plural, singular is fungus. They use organic chemicals for energy, such as glucose. The fungi include the multicellular molds shown here, and the mushrooms, which are also multicellular, and the unicellular yeast, shown here. Most of the fungi are decomposers, but a few of them are pathogens, like Candida albicans is a human pathogen. The kingdom Protista can be broken down into 3 different groups. There are the plant-like protists, and they include the algae. There's the animal-like protists, and they include the protozoa, or they are the protozoa. And then there's the fungus-like protists, and we don't have a term for that. Water molds are an example of a fungus-like protozoa. If you don't know what a water mold is, it was what caused the potato blight in Ireland, making the Irish starve to death, and that's what brought a lot of the Irish to America. And I don't remember if that was in the 1800s or the early 1900s, but sometime around then. The algae, all of them are photosynthetic, so they get their own energy from sunlight and then can convert the energy in sunlight and CO2 and water into glucose. They do produce oxygen as a byproduct, as well as they produce glucose. The protozoa include the amoeba, the paramecia, and the euglena. All of these either absorb or ingest organic chemicals, so they need to absorb or ingest their food. The protozoa may be motile. And if they're motile, they can be motile by flagella, which is the only way that bacteria and archaea are motile from flagella. They may be motile by cilia, and they may be motile by pseudopods. A pseudopod is where the organism, if it's moving that way, it puts down the cytoplasm here and then pulls up the cytoplasm here. Some of the protozoa are parasites, so we will talk about them a little more in this class, but not so much today. Any question about the kingdom Protista? If not, let's move on to the kingdom Plantae. You can divide the plants into the mosses, the ferns, the conifers, the flowering plants. All plants are multicellular. We're not going to be studying plants in this class because, once again, plants do not cause human disease. The animals can be divided into the sponges, the worms, which are helminths, the insects, and the vertebrates. All of the animals are multicellular. We will talk a little bit about the worms or helminths because worms and helminths can cause disease. And this class will be focused on organisms that cause human disease. We will not talk further about sponges and vertebrates, and barely we'll talk about insects. All right, any question about the classification of microorganisms? Classification of life, actually, because most eukaryotes aren't microorganisms. All right. What did I want to say about classification? Lost it. I'll try and think what the heck I wanted to say, but totally lost what I wanted to talk about. One thing I want to mention is that humans call numerous animals worms. In reality, there's 3 different types of worms, 3 different families, and they are as different from each other as they are to humans. So even though we group them as worms because to us they look like a worm, they're not genetically similar. And the 3 families are the annelids, like earthworms, the flatworms, or platyhelminths, and then the nematodes, or the roundworms. And genetically, they're not any closer to each other than they are to humans. So I'm just warning you that the helminths, we probably shouldn't lump them together, but humans have looked at them and said that's a worm, and so that's sort of what we continue doing. All right, we've talked about microbes and microorganisms. I'm going to now talk about what comes under the umbrella of a microorganism. When we're talking about a microbe, what do we mean? We mean all bacteria, it's a microbe, all archaea. We also mean viruses and prions, but these are special because viruses and prions are not organisms. They do not have cells. So I will call them a microbe. For my own use, I don't call them microorganisms because they're not an organism. But some authors will call them a microorganism. Some of the eukarya are a microbe. All of the protozoa all of the yeast, the molds, some of the algae are microbes. If it's microscopic, it is a microbe. Some of the algae, like the kelp that wash up on the Oregon or Washington beaches, is not a microbe. It is a living structure, and we do group them as algae. So some of the algae are microbes and others are not. And then the helminths, we do consider them a microbe, even though the adult worms are not microscopic. As I mentioned, the adult tapeworm, or the human tapeworm, sorry, which is normally called the beef tapeworm, but it is found in, well, let's not go into that. It can be 30 meters long. So why are we grouping that in the microorganisms? Well, of the helminths, of the worms, even though the adult can be very big, like 30 meters is the biggest, I think, the infective stage of the worm is usually, if not entirely, a microbe. For many of the worms, it's the fertilized egg, which is the infective stage. But there are some which are very small microbe, which can penetrate through, burrow through the human skin, and it is not a single fertilized egg. It's a microbe which can actually burrow through the single skin. The point is that all of the helminths, the infective stage is a microbe. And then the helminths also cause human disease, and microbiologists have studied organisms that cause human disease. And the parasitic worms do cause parasitic disease. So that's why microbes do include helminths. Any question about any of that? All right, I've already mentioned that of the helminths, only the flatworms and some of the roundworms cause disease. So only some of them are parasitic, at least of the roundworms. Let me think about the flatworms. I think all of the flatworms are either parasitic to humans or animals. That's not true. Not all flatworms are parasitic, but of the parasitic flatworms, all of them cause disease. So if I word it parasitic flatworm, that's correct, but not all flatworms cause disease. All right, any question about any of that? So you'll notice that there's two worms, the flatworms and the roundworms, which microbiologists study and consider a microbe. The annelids, like the earthworm, do not cause human disease, and microbiologists do not study them. I wonder if they're a microbe. I'd have to look that up. I don't know. Any question on any of that? All right, let's briefly talk about viruses. I mentioned that viruses lack cells, so we call them acellular. Viruses do have a DNA or RNA, which is the centralmost molecule, and their genome. But their genome could be either DNA or RNA, never both. They have that genome surrounded by a protein coat. The protein coat may be surrounded by a lipid envelope. Viruses only replicate inside their host cell. They do not replicate outside their host cell. In fact, they are not metabolically active when the virus is outside its host cell. Viruses are not part of the three-domain system, mainly because they're not cellular. Any question about viruses? We will talk much more about viruses late in the term, but for now, this is just an introduction. When we look at the three domains of cellular organisms, the bacteria, the archaea, and the eukaryotes, we can see that the bacteria and the archaea have the cell type prokaryote, and the eukaryotes have the eukaryotic cell type. When we look at the chromosome, the bacteria have one chromosome, the archaea also one chromosome. The eukaryotes have many chromosomes. There's a difference in the chromosome between the bacteria and the eukaryotes. The bacteria have a circular chromosome. That means the bacteria is in a circle, so that the starting place is the same as the ending place. Eukaryotes have linear chromosomes, like the X chromosome. It has a starting place up here, and then an ending place down here. So it's not the same. We'll talk about histones and introns later, but eukaryotes have histones and introns. Bacteria have no histones, and they have no introns. When we look at the bacteria and the eukaryotes, we notice they have a difference in the ribosomes. The bacteria has a smaller ribosome at 70S. The eukaryotes have a larger ribosome at 80S. The archaea has a 70S ribosome. When we look at the cell wall of bacteria, the main molecule is peptidoglycan. There is no peptidoglycan found in the archaea or in the eukaryotes. All right, any question about any of that? Let me check on my schedule here. Yeah, I'm going to try and work a little further because we only have a little bit of next class to work on chapter 1, and we have a lot of slides to go yet. Where is that? When we're talking about the history of microbiology, it largely is concerning the debate over whether spontaneous generation gave rise to microbes or whether a microbe could only come from another microbe. The history of microbiology also includes the golden age of microbiology. You don't need to know the years, but it's when microbiology first got started, and whenever people looked at something, they discovered something new. So it's called the golden age. And there we're looking at the history of microbiology, the golden age, and then the later age. We'll talk about a number of those things here. When I'm going through the history of microbiology, I'm going to give you a date for things, and that's just to sort of organize things. But of the dates, you don't need to know any of the dates except for this first date. Over 3.5 billion years ago, we can find in the rock, in the fossil record, ancestors of the first bacteria that were the first life on the planet Earth. And here we're looking at a fossil, and that's an artist's rendition of the fossil. And there's another one. So that's the only date you need to know of all the history of microbiology. You should know that the Babylonians first wrote about microbes, and that's when they were writing down the recipes for making beer. They wrote down about microbes. Of course, they did not know that microbes were making the beer because they could not see the microbes. And then later, the Mayans wrote down recipes for making a fermented beverage from cacao that we would call chocolate today. And when I'm going through these things, you should realize that before the Mayans could write down how to make chocolate, there first had to be the Babylonians writing down about microbes. And before that, you have to know that there was a fossil record that dated to about 3.5 billion years ago. So you do need to know the order of the events in these history of microbiology, but you do not need to know the dates, except for this first one. Any question about any of that? All right. The first observation involved in the history of microbiology, you don't need to know the date, but in 1590, Janssen developed the compound microscope. Without the microscope, we could not see microbes. And so in 1590, the first microscope was invented. This is actually before the history of microbiology, because there was no microbiology until the microscope was invented. But nobody used the microscope to look at cells until 1665, when Robert Hooke reported that living plants are composed of little boxes that he called cells. Let me see if I can get this. Oh, here it is. Let me quickly draw this. He saw, bring that across. In the tissue, like in a shaving of cork, he saw cells like this. And he called them cells because this reminded him of the cell in a monastery where the monks lived. There'd be a hallway out here, and then a little door right here. And then this is the room the monks did whatever the monks did in the room. Obviously, they slept there, but they probably prayed there and did other things that the monks did in their room. And the monks called this a cell. And so this looked like a cell to him. So he called it a cell. And that's where we get the name cell. Any question about any of that? Now, Robert Hooke did look at animal tissue, but he could not see cells. And that is because animal cells are bounded only by a thin cell membrane. And in the primitive microscopes of his day, the microscopes were blurry, you couldn't find focus, and nobody could see cells in animal tissue because the microscopes just weren't good enough. But in plants, because they have a thick cell wall, people could see the cells. Any question about any of that? Anyways, Robert Hooke seeing cells in plants gave rise to the cell theory. He didn't give rise to it, but it developed out of his finding that all living things are composed of cells. And then about 100 years after Robert Hooke saw cells in plants, the microscopes got better, and people could finally focus in on animal tissue and then see that it was composed of cells. The cell theory was actually written up and proposed by Virchow much later, and we'll talk about it in this class. The above things are not specifically microbiology, because Robert Hooke was not looking at a microbe, he was looking at plant cells. And the plant, of course, was a very big tree. He looked at the shavings of cork, which is the bark of the cork tree. All right, I think I'm gonna stop here. And we will pick up here next time. Any questions about anything before we go? Let me make sure that I'm supposed to go, but I think I'm supposed to go at 6:20. Yep. So I'll see you at 6:30 for the lab. Any questions? All right. Bye.